Hello, my name is Paul Goulding and I'm a Business Development Manager for Progenesis QI. My talk today is entitled The Statistical Power of Co-Detection for Discovery Omics Analysis using Progenesis QI software. The topics I'll discuss today include, firstly, what is Discovery Omics Analysis and why do we do it? I'll then talk about the nature and complexity of the data and the challenges this presents when we try to analyse it. I'll then describe the Progenesis solution to these challenges, which we call co-detection. And finally, I'll summarise the main points. Omics is a term used to describe many types of analysis, but the particular type of omics analysis I'll be talking about today can be applied to small molecule metabolite ions in metabolomics, lipid ions in lipidomics, and peptide ions from protein digests in proteomics. For simplicity today, I'll refer to all these types of analytes as ions. This slide comes from the National Cancer Centre, and it's their suggested workflow for biomarker discovery and validation. Discovery analysis is the first phase on the left, and is used to find biomarker candidates that can then be passed on to the validation stage. The discovery phase involves analysing all detectable ions to see what is being differently expressed between the experimental conditions, such as control versus treated or healthy versus diseased. Since this can involve comparing many thousands of ions across perhaps dozens of samples, the complexity of the analysis is huge, as we'll see later. In Discovery Omics Analysis, we're trying to find relatively small numbers of expression-changing ions amongst many ions that are not changing. A simple analogy is a spot the difference puzzle, in which the two pictures are mostly the same, but there are just a few differences, such as a missing branch of the tree, a missing bird, a missing building, the direction of the man's crossed leg, and a missing gap in the paving. But in Discovery Omics Analysis, we also have to take account of biological variance amongst our samples. So we must run multiple samples in each condition and look only for consistent or statistically significant differences between the conditions, of which there are now only two, the building and the tree branch. If we now look at the nature and complexity of Discovery Omics data generated using LC-MS equipment, it's composed of two separation elements, liquid chromatography, or LC, and high-resolution mass spectrometry, or MS. Each of these alone can produce complex separation patterns. So when we combine them, we are essentially multiplying one by the other to generate data of huge complexity. However, it is possible to visualize this data by combining the one-dimensional separations into a two-dimensional ion map, in which the ion signals appear as small dots in characteristic patterns. Actually, the 2D ion map has a third dimension, which is the mass spec intensity. And if we zoom in on a single ion signal, we can visualize it in 3 dimensions. And in this way, you can see how it's possible to quantify the ions by measuring the 3-dimensional volume of the isotope peaks and adding them to give the ion abundance. In Discovery Omics Analysis, we are then comparing these ion abundances between 2 or more conditions or groups of samples. But in order to do this, we must be able to match the same ions across the different samples. Otherwise, we might find ourselves comparing different ions, which would give meaningless results. In this view of our control and treated samples, it doesn't seem as if this should be a problem. After all, the patterns of data on both samples look very similar. However, if we zoom in on the same area of each sample, things no longer look quite so easy, since the zoomed-in patterns look quite different. What's worse, the conventional method of analyzing this type of data is very inefficient. In this method, the peak picking is performed independently on each sample. So in this case, it would detect the peaks on the control sample, followed by the treated sample, and then on all the other samples in the experiment, one by one. The result is that very different detection patterns are produced for each sample, making matching of the ions between the samples very difficult. Let's see how many ions we can match between our control and treated samples. To help with this, we'll first apply some retention time alignment so that the ions which are clearly the same are aligned in the vertical LC dimension. We can then see that a certain number of ions can be matched between the two samples. But there are also a lot of ions that are either detectable on only one sample or have been detected differently on each sample and therefore cannot be matched for comparison. What's worse, in this example, we're only trying to match a few ions between two samples, while in a real experiment, we may be trying to match thousands of ions across many samples. So the problem is many times more complex than shown here, and even here, we cannot do it well. This inability to match ions across different samples affects the statistical reliability and statistical power of the experiment, as shown in this table. Here we have a simple 3 versus 3 Discovery Omics experiment, with 3 control samples being compared to 3 treated samples. Each row of data represents the measured abundances of the same ion across the 6 samples. In the second row, there is a full set of data, 3 control abundances compared with 3 treated. So in this case, we can use a t-test to calculate a reliable p-value, which is the probability that there is no expression change occurring. However, in the other rows, we have gaps in the data, which occur when we cannot find the ion whose abundance we've measured in the other samples. These missing values mean we have to calculate the p-value using fewer data, which means they cannot give us a reliable probability for an expression change. It's possible, therefore, that we could miss some expression changes which are there to be found in the data, but we cannot see them because of the inefficient way we are analyzing it. What's worse, if we take the obvious route to improve our experiment by running more biological replicate samples, we get more missing values, since it's harder to match an ion across 12 samples than it is across 6. We therefore have to increase the number of samples by a lot in order to get a relatively small increase in the ability to find expression changes in our data. So what if there was a better, more efficient way to analyze Discovery Omics data? Well, Progenesis QI software has a solution which involves a workflow of data alignment followed by co-detection, which results in 100% matching of all ions across all samples and no missing values. But how does this work? Well, here we have a simple 3 versus 3 Discovery Omics experiment with conditions A and B to compare. If we zoom in on the same area of all 6 ion maps, we can see more detail of the individual ions, including the isotope peaks. The first step of the workflow is to align the data before any detection occurs. To do this, Progenesis QI automatically chooses one sample as the alignment reference. In this case, it's chosen A1, and all the other samples are then aligned against it. The first step of alignment is to overlay the samples. You can now see the data from the 2 samples in 2 different colors, and that they are currently misaligned in the vertical LC dimension due to small retention time differences between the samples. To compensate for this difference, Progenesis creates a series of alignment vectors, which are then used to align the data. So, where we clearly had 2 sets of data, we now appear to have just 1. We can then separate the samples and repeat for the other 4 samples. Then the aligned samples are combined or added together to form what we call an aggregate sample ion map, which is a composite of all the individual sample ion maps. Progenesis then performs peak picking on this aggregate image. Since the aggregate includes all the ions present in each individual sample, this creates a detection pattern which, when we separate the samples, can be retained to give the same detection pattern for all the samples in the experiment. This means it's now easy for Progenesis to find and compare any of these ions across all the samples. Here we can see the ion co-detected on all samples, and quantitative analysis results in a relatively high p-value, indicating no significant expression change for this ion. This is confirmed by the expression profile, in which the ranges of quantities in each group are large and overlapping significantly, and by the ion view for each sample, which all appear to be of very similar intensity. In another example, the p-value is much lower, indicating a significant expression change with a fold change ratio of 4, which is confirmed by the expression profile, in which the ranges of quantities in each group are very small compared to the difference between them. And here, the p-value is extremely low, and the fold change ratio is infinity, indicating a presence-absence situation, which is confirmed not only by the profile, but also by the ion views, which clearly show something present in Group A and nothing in Group B. So, the benefits of the co-detection workflow are no missing data in the stats analysis, since all ions are detected and quantified across all samples. This means more reliable statistical analysis, and therefore more chance of finding the expression changes that are present in the data. Peak picking is also more reliable and sensitive, since it's performed on the aggregate ion map, which has superior signal to noise compared to any individual sample ion map. And finally, normalisation of data across samples is also more reliable, since it uses exactly the same ions in every sample. The more efficient is normalisation, the more chance you have of finding the expression changes in your data. So to summarise my talk, Discovery Omics Analysis is a key stage in biomarker discovery workflows, but the complexity of the data makes it very challenging, particularly in matching thousands of ions across many samples in an experiment. Progenesis QI has a solution to this problem, which we call co-detection, and this enables Progenesis to find more expression changes from the same data than would be found by other software. This is because co-detection generates no missing values, enabling more efficient statistical analysis. It also has more sensitive and reliable peak picking because it's performed from the aggregated data, and it enables more reliable data normalisation through using the same ions on every sample to normalise. It just remains for me to thank you all for your attention. I hope this is giving you further insight into the potential problems of Discovery Omics Analysis and the Progenesis solution. Thank you.